What's up, nerdlings? Today I'm going to be discussing gene regulation in prokaryotes, so get ready for some operons. So first of all, you might be asking, but Queen Nerdling, what is gene regulation? Well, it is the process by which organisms induce or turn on or silence and turn off gene expression. You might also hear the terms upregulation or downregulation of genes. Those are two different terms that can be used interchangeably. So induce, on, upregulate, silence, downregulate, off. So why is gene expression so important? Well, I'm glad you asked. So all organisms have to maintain homeostasis in order to function properly. So what helps this is protein expression, and that's going to be dependent upon what the cell needs, as well as how it's responding to its environment, both internally, meaning inside the cell, and the external environment itself. So one of the examples that most of you are probably familiar with is insulin expression. So when we have high blood sugar, insulin and insulin receptors need to be expressed or turned on. When we have low blood sugar, insulin and insulin receptors don't need to be expressed. So in eukaryotic organisms, we have all of these different types of cells that possess identical DNA. But each of those cells might contribute to a different function because they're part of a different tissue that forms a different organ, which forms a different organ system. So this means that although every cell has all of the DNA it needs, that certain cell types need to turn certain genes off permanently or have certain genes turned on permanently in order to function correctly. So for example, we don't really want our heart muscle cells to be producing adrenaline. So these genes need to be silenced or downregulated or turned off. So gene expression is slightly different in prokaryotes and eukaryotes. Today we're going to be mostly focusing on prokaryotic gene expression, but just to kind of give you an example, prokaryotes don't possess introns or a nucleus. And so if you do a flashback to protein synthesis, whenever those messenger RNA strands are being synthesized in the nucleus, which prokaryotic cells also lack, we have a spliceosome, which is actually going to splice introns together and splice exons together. The exons get to exit the nucleus, while the introns remain inside of the nucleus. So exons exit, those then get sent to ribosomes where they are upregulated, meaning those genes are expressed, and the introns stay inside the cell and they are not expressed. So because prokaryotes don't possess introns or a nucleus, transcription and translation are coupled together, meaning they're happening at the same time. There's no separation like there is in eukaryotic gene expression. Eukaryotes, on the other hand, they do have introns and exons and a nucleus. So transcription and translation are not coupled together, meaning there is a temporal space between those two processes. These differences in protein synthesis cause prokaryotes and eukaryotes to have extremely different methods that they employ to regulate their gene expression. So let's take a look. So prokaryotic gene regulation. They regulate gene expression by controlling the transcription of genes. And they do this because transcription and translation are coupled, meaning there's no way to prevent the messenger RNA from being translated as soon as it's made. So prokaryotes accomplish this by having sets of genes arranged in a structure that's called an operon. So operons are transcriptional units that control the expression of a gene or a set of genes that are found only in prokaryotes. So operons consist of the following parts. They have a promoter sequence, and this does not code for a protein. This is where RNA polymerase binds to initiate transcription. So the promoter sequence is basically like, hey, yo, RNA polymerase, this is where we're starting. And so the RNA polymerase knows where it's going to start. And that's going to be this section right here, the promoter region. So we then have an operator sequence. Again, this isn't going to code for a protein. It's part of the promoter sequence. And this is where repressor proteins bind to prevent transcription. So we're going to get into that in the next few slides, but we need these repressor proteins to keep the transcription of this gene off or remove them to turn it on. So we also have a regulatory gene sequence, and this codes for the repressor protein that binds to the operator sequence to prevent the transcription. And then once you pass that region, or this little control region, and transcription actually does start to occur, you have the genes that are actually going to be transcribed. And that is what's actually going to code for a protein that's going to be expressed. So operons are transcriptional units that control the expression of a gene or a set of genes, and they're found in prokaryotes only, and they are composed again of the following. So the structural genes, again, they're going to be up here, and they're related and used in biochemical pathways. That promoter, remember, is going to be like, hey, yo, RNA polymerase, let's go right here. Start here. So that's the nucleotide sequence that can bind with RNA polymerase to start the transcription itself. And it occurs, this sequence also contains that operator region that I spoke about in the last slide. The operator is going to be the nucleotide sequence that can bind with the repressor protein to inhibit transcription. And remember, inhibit means stop. So the regulator gene. This gene produces a protein called a repressor that can stop or inhibit the transcription of an operon by attaching to the operator. I know all of these terms seem super confusing, and I'm spouting out a lot of different terminology. So you might just want to take a little bit of time, pause it, go back, and kind of get used to some of the terms that I'm using. So again, regulator gene produces a repressor protein. And the repressor protein inhibits or stops transcription from occurring. So right here, this is where the repressor is going to bind into the operator region. So when this is being repressed, RNA polymerase sees the promoter. Promoter's like, yo, RNA polymerase, come on over, let's get started. So RNA polymerase is like, I'm going, what? And it stops because there's a repressor protein. When there's a repressor right here in the operator, that RNA polymerase, it can't work. It can't start transcribing. So it's basically waiting there like, well, I showed up, but nothing's going on yet because the door's locked. So it's almost like an inhibitor and an enzyme. So when it's occupying the operator site, RNA polymerase can't come along and transcribe that gene so it can be upregulated and expressed. So repressors have allosteric properties, just like in enzymes, they have an allosteric site that changes the conformation of the enzyme. So it changes the conformation, meaning it changes the shape. So modulators can bind to the repressor at an allosteric site and change the conformation or shape of the repressor. That is going to activate or deactivate the repressor. Usually the modulator is a product in a biochemical pathway. So right here, we have our regulator protein, and this would be our little inducer. When it binds with this allosteric site, you see there's a slight conformational change. So a straight binds, and all of a sudden, we have a conformational change. So similarly, we can have a regulatory protein where this is constantly on, and when it binds, it turns it off. So we can have inducible and repressible operons, and we'll get there shortly. So the two different types of operons that we're going to be going through are called inducible and repressible. Inducible operons are basically always off or down-regulated. So they're always turned off, and they're waiting for something to turn them on. So they have the repressor protein in their site, and they're waiting to be turned on. So they have to be induced. whereas repressible operons are always on. RNA polymerase, that was going, we're synthesizing stuff, yeah, until something comes along and inhibits or stops it. So inducible operons are always off. Polymerase is like, I'm waiting, still waiting, until eventually they're turned on or induced. And again, repressible, they're always on until something has to shut them down. They're like, RNA polymerase, this party's going too long. We're going to shut you down. Stop. So inducible, like I was saying, they're normally off and they have to be turned on. So in normal conditions, there's no inducer molecule present. So they're constantly going to be off. So the repressor protein is constantly bound to the operator sequence. So RNA polymerase is just waiting. And this prevents the transcription of the gene. Induce the molecule concentration increases. So the inducer binds to the repressor and inhibits it or changes its shape. The repressor can't bind to the operator sequence anymore, and then the RNA polymerase is allowed to transcribe that gene. So again, an inducer molecule, that's a little typo right there, the concentration increases. So for example, if I'm going with my milk example, so I'm going to consume milk. Milk has a specific sugar in it called, if you said lactose, you are correct. Now there is a specific enzyme that breaks down the sugar in milk of lactose, and that enzyme is called lactase. So whenever we consume milk, we're going to have lactose in our bodies. And we're also going to have something called allolactose. Now that is actually going to bind with the repressor protein. Now when that happens, it's going to change the conformation. That repressor protein is going to be removed, and RNA polymerase can then transcribe what's going to code for the lactase enzyme. So we can break down the milk. Now, because it's an inducible operon, it means it's always turned off, because we don't always need to be making lactase enzyme unless we've consumed lactose. So it only gets turned on when it's needed. So here's an example of the lac operon. Again, it's an inducible operon, meaning it's always turned off until we need to use it. So right here, this would be our allolactose inducer, meaning this is something that I've ingested when I've drank milk, eaten cheese, eaten ice cream, now I'm hungry. It's going to bind with that active repressor protein. When it gets bound to that active repressor protein, it's going to be removed from this operator region right here, which is going to allow RNA polymerase to transcribe all of those genes, which eventually will code for lactase, which will break down the lactose and allow you to use the sugar in the milk. So whenever it's repressed, the lactose operon or the lac operon is going to look like this. So it's actively being repressed, and this is the normal state, meaning unless we consume dairy, it's always going to be turned off. So RNA polymerase is constantly waiting. It's like, I'm gonna drink some milk, I'm gonna eat some ice cream, I'm waiting. But until that happens, it is blocked by this active inhibitor. Now again, if we consume milk, we're going to have allolactase that comes and binds with that active inhibitor or active repressor, and eventually it's going to be removed, and then RNA polymerase will be able to transcribe the gene. Whenever we're not consuming dairy products or milk, that's going to be turned off, and RNA polymerase is not going to be able to transcribe those genes that follow. So when it's turned on, we have the allolactose. So say I ate a big bowl of cookies and cream ice cream. That's what I really want right now. So I have all of this lactose floating around in my bloodstream. That's going to eventually bond with the repressor protein. That repressor is going to have a conformational change and be removed from the operator region. And now RNA polymerase, as long as I'm not lactase persistent, is going to be able to transcribe the lactase gene and give me lactase enzymes so I can break down the sugar in the milk and utilize it in my body. So when the lactose runs out, then the repressor protein is going to go back into the active state and stop that gene from being expressed. So as far as repressible operons go, those are always upregulated or turned on, and they have to have something that turns them off. So under normal conditions, there is no co-repressor present. So the repressor protein is in the wrong shape and it is inactive, meaning it's always basically off. So it cannot bind to the operator sequence because it's not in the correct conformation or shape. So RNA polymerase is constantly able to transcribe the genes. Now, co-repressor molecule increases in concentration. When the co-repressor binds to that repressor protein, it's going to change the shape into an activated shape. The repressor protein can now bind to that operator sequence and stop RNA polymerase from transcribing that gene. Now, the example that I'm going to go over is called the trip operon. So you may have heard of tryptophan around Thanksgiving time when you're eating a lot of turkey because it's high in tryptophan. Well, tryptophan is actually an essential amino acid that we need to use in our body constantly. So our body is constantly synthesizing tryptophan. Now, if we consume a big meal that includes a lot of tryptophan, like our Thanksgiving dinner, if we eat a giant turkey leg or something like that, we're going to have so much tryptophan in our body that it's going to bind to that repressor and it's going to basically activate it, so it will come into the operator site and stop RNA polymerase from transcribing the genes to create tryptophan because we have enough of it. So the repressor gene, or the TRPR, the promoter and the operator sequences, and then the structural genes of E, D, C, B, and A are all going to code for that tryptophan. So when the trip operon is active, it's constantly synthesizing tryptophan, so making tryptophan, until something represses it, like we eat the turkey leg. Now, when we eat the turkey leg, because we already have a lot of tryptophan in our body, the tryptophan co-repressor is going to bind with the inactive protein, thus activating it. So again, conformational change. Once it's activated, it's going to bind at this operator region, and again, it's going to stop the RNA polymerase from transcribing those genes because there's enough tryptophan in our body to be able to use for the synthesis of different proteins. So we don't need to keep making it until the level decreases. And once that level decreases, the co-repressor for tryptophan is going to break off, and this will become active again, and our body will again start to synthesize tryptophan. So remember, the trip operon controls the expression of proteins that synthesize that special amino acid, tryptophan, and it will be used for protein synthesis. So eventually, most of the tryptophan will be incorporated into proteins throughout our body. That's going to lower the concentration, and that will signal to the trip operon to start making more tryptophan. So I hope that was helpful and that you enjoyed your journey through the trip and the lac operons. Stay tuned for eukaryotic gene regulation. I'll see you guys soon.